Okay, so continuing on with the classification, we've seen so far archaea, we've seen bacteria. So now we enter the third phase of the organisms that we're interested in studying in this subject, which is going to be Protista. So specifically, we're going to look at protozoans, so the ones that are hailed to be animal-like per se. And so this one overlaps fairly well with the laboratory. So you're going to see me talking about a lot of the examples that we have here and there, and I'll show you some of the cool examples that are pretty gnarly and kind of interesting in my humble opinion. So that being said, since we're going to be looking at protists and protozoans, more anything else, let's make sure that you're familiar with the stuff that we learned in topic C, which is obviously that these are all eukaryotes. That means that they do have most, if not a lot, of the organelles. And in this case, since they're not like bacteria, they do not undergo binary fission or anything like that. Instead, they're just like any mammal or humans and things like that. They divide by mitosis, and some of them have meiotic capabilities. And so they're related relatively to all four other eukaryotic kingdoms that we've discussed in the past. So our protozoa, specifically the ones we are looking at, are all going to be unicellular. And that means also that, very similar to us, they do not have a cell wall. And probably one of the most critical things in microbiology altogether, not even just these organisms, but micro altogether, is that these all require water. More than anything else, it requires some sort of liquid or moist environment to move. So that means most of the time, you'll find these in bodies of water. So whether it's your lake, your ocean, your pond, your puddle, your glass of water, your pool, anything that has some sort of body of water, you'll find them. And then technically this will also count in things that are relatively humid or moist, like soil. So as long as it's not fully dried up. So as long as there's a little bit of water in your dirt, that counts as well. Now, the variety behind these guys is also pretty massive, but a few of them are pathogenic and either useful in some sort of industrial or medical purpose. So we'll probably look at most of the ones that are pathogenic that are interesting for us in micro, and probably give you a couple of examples here and there on the fun and cool ones that we know. Now, overall, they're pretty much, in terms of their cellular structure, they're similar to most other eukaryotes, and pretty much more than anything else is that in terms of their nucleus, in terms of the DNA that they do have quite a bit of variation in terms of how many copies do they carry. So like humans and a couple of animals, we have two copies, one from mom, one from dad kind of thing. Similar to plants, possibly they can have multiple copies of the exact same chromosome. So there will be some variation in there, including the size of their nucleus and the shape of it. Similarly, in terms of their mitochondria, you'll see a lot of variations in terms of shape and what they do. And then lastly, sometimes some of the organelles will be present or absent. One of the more infamous of them all is the vacuole. Some of them that are kind of almost plant-like will have vacuoles, some of them will not. But one of the key features that we see here that we're interested in, and this is probably by definition something you should know for the exam, is that they all are motile, meaning that they all move, and we classify them by that motility. Now, the key feature here is that virtually most of them move, except there is one group that doesn't. So technically, we still classify by the fact that they do not move. So keep that in mind. And so they move by all basic means that we've talked about in the cell lecture, whether they have cilia or flagella, or parts of their bodies kind of extend and protrude, called pseudopodia or pseudopods. So they all more or less move, swim, crawl, or some sort of variation thereof, with the exception of one of those classifications, just so you know. Now, part of that motility is what indicates kind of one of their stages in life, called a Trophozoite Stage, and there's some variations of that terminology too, in case you're wondering. And so that's pretty much the active stage. That's the part that is going around looking for food. So anytime you see them swimming, they're probably trying to kill you. However, they also have a kind of the converse stage in which they're pretty much not doing anything. They're growing or developing, or they're doing something else. and that's a resting stage referred to as a cyst. Now, in terms of what they're trying to eat, most of them are like us. They're chemoheterotrophs, meaning that they're looking to eat other things that are usually organic to supply themselves with whatever they need in terms of nutrients, macromolecules, and so on and so forth. And then some of them, very similar to our group of fungi, are saprophytic, meaning that they specifically go after things that are dying or decaying. And like I said before, some of them have similarities to plants, similar to algae more than anything else. And these are referred to be photoautotrophic, and we'll clarify those as we go. Now, some of these, in terms of the reproduction, like I said, it's very mitotic and meiotic. So they can have an asexual type of division. So that would be more like mitosis, not like binary fission, in case you're wondering that. Okay. And then more than anything else, they have these cool variations on how they do that in terms of how they share and trade materials if they're doing a sexual version of them. And so they produce, very similar to us, like we produce sperm and eggs kind of a way to reproduce sexually, they produce counterparts to those as well. And so these are referred to as gametocytes. So we'll start kind of highlighting which ones do which and where and why as we go through the examples. So that's part of the plan of what we're going to do today. Now, here's where it gets really, really interesting, because great updates in our current understanding and research have kind of led us to understand that the classification that used to exist, used to be just 4 of those that everybody was familiar with, has gone up. And so now there's currently about 7 different taxa that are being recognized based on their ribosomes. But there's about to be this new reclassification about them, in which they're about to be split up into much bigger groups. Now, we're going to stay with what we know right now before it changes again. Just be aware that there is this possibility that things will need to be updated a little bit more. So keep that in mind. Now, the original 4 that your book even lists are based on what those old kingdoms that we had talked about before. So the idea of protozoa fell under 4 groups called Sarcodina, Mastigophora, Coelophora, and Sporozoa. Those have been all reclassified. And so even in your lab manual, because it was based on this material, there were several phyla that existed in there, kind of the Dinoflagellates and the Coelophores, that no longer exist in there too, and they've been reclassified as well. So it makes it a little bit confusing sometimes to kind of keep up with this material. So I'm going to try and just stick to what we know, what we have so far, what is currently up to date, let's put it that way. And so that's what we're going to learn for both the lab and the lecture. So that means that we're going to have to update some of the material we have in there. So this is probably where you want to kind of pay attention to the lecture more than anything else, since this is kind of going to be what we have in the exam itself. Okay, so if you look on the right-hand side, we have this all material in orange. Remember that that highlights the actual taxonomy. And then I'll indicate which ones are phyla, which ones are classes. You'll usually see that in the cool purple color. And then the currently recognized material that you need to know, okay? So for the moment, what you need to know is the following groups. First group is called Parabasala. Second group is called Alveolata. Third group is called Cercozoa. Fourth group is called Radiolaria. Fifth is Amoebozoa. Sixth is Euglenozoa. And seventh is Diplomonadida. So those are the current 7 that we are using. This is what we're going to be focusing on both the lecture and the lab. That's what you're going to be tested on. However, one of the things that did happen very recently, and then this one has been already established, is that a brand new phylum has been added. Not that I'm going to test you on this, so don't worry about it, hence why I have it in this color, is that they're going to be fusing Parabasala and Diplomonadida in this kind of superphylum that is about to pop up. So keep that in mind, that this is what's going on right as we speak. Now, kingdom Protista is considered to be what we call polyphyletic. That's a kind of a very evolution and scientific term that we use that more or less means that this group of organisms is derived from more than one common evolutionary ancestor, or at least in some sort of ancestral group. And so therefore they're not really suitable for being placed into individual groups. They're all related together. So rather the groups that you're seeing here, this is as close as we can get them, but it doesn't necessarily mean that they're directly related, especially in the way that I have them ordered as well. So keep that in mind. So this is kind of a collection of slightly unrelated organisms that all fit the category of motility, unicellular, lacking a cell wall, and so on and so forth. So that's where we're going to go with this. Okay. So what do we want to know about these guys? Well, in terms of their reproduction, they have this kind of almost alternation of generations kind of thing, for those of you who remember plants. And this has to do with the fact that some stages of their reproduction, that means their life cycles more than anything else, there's a stage in which they're kind of always going around in this trophozoite stage and then more or less reproducing. And the cells themselves are what we call haploid, meaning very similar to a sperm or an egg, so half the amount of material. And for a very, very short time, they kind of fuse together and become diploid, kind of like us, fusing an egg and a sperm, more or less. And that's what we refer to as a haplontic life cycle or life stage. Conversely, there could be organisms that live as diploids like we do. We're already a combination of both of our parents. And then at some short stage of their life, they produce gametes that are the half cells. Now, what's interesting about that is that that wouldn't be very useful until we discover the third version of it, in which they can go back and forth, kind of alternating between a diploid and a haploid stage, and just kind of going about their life kind of crazily. So this gets a little complex. Now, this is the only time I'm going to highlight this, simply to kind of make you understand that these organisms are very, very variegated in terms of how much, in terms of their reproduction, motility, and even cellular structure, there's all this crazy variation that we need to know. So we're just going to do the highlights. So just keep that in mind. Don't be overwhelmed. All right. So we're going to go with the same plan that we've gone before. So we're going to look at an organism or at a phylum and specifically discuss their properties that are highlighted in blue. I'll give you the taxon or the taxonomy that you need to know, which is highlighted in usually green and in orange. And then I usually give you some sort of examples, pretty pictures, that kind of thing, for you guys to be a little bit more concrete in terms of the material that you want to know. Okay. So starting off with the right, so you'll notice that the classification is fully listed on the left. Okay. We have our very first phylum, which is Parabasala. Now, the term Parabasala comes from something that is derived called a parabasal body, which is the equivalent of a Golgi apparatus. So that's where that name kind of derives from more than anything else. Now, what's unique about them also is that they also lack a mitochondrium. Now, that doesn't mean that they don't produce energy. That's not usually what it means. What it means is it can use its own plasma membrane as a way to derive its own energy. So it uses its own pieces rather than a separate organelle to do so. Okay. Now, out of there, one of the reasons why we pay attention to specifically Parabasala is because of two particular organisms that are kind of cool to know. Probably the first one that is infamous of these all is Trichonympha, which is also part of the organisms that help lots of things like termites and certain ruminants in digestion of complex polysaccharides. So that's where that guy kind of usually derives. And you can see him here a little bit further developed. There it goes. However, probably the more infamous of them all is the one that is involved in sexually transmitted infections. And so this is Trichomonas, often known as trick, believe it or not. And probably the reason behind it is because the term trick usually is the Greek term for hairy. So that's where kind of you can see all these little cool little extensions sticking out like it had a little wig. And more than anything else, probably everybody's familiar with this simply because of the STI that is associated with it. And this is known as trichomoniasis. So it's a very, very commonly sexually transmitted infection or disease, if you will. And so its infamous name behind is called Trichomonas Vaginalis. The symptoms kind of vary depending on how, the intensity of the actual infection can be, and it's very, very easily transmitted. So usually per year, about 1% of our United States population usually has this. So that's a pretty good group out there. Now, about 70% of those people that are infected, interestingly enough, are also asymptomatic or don't really report any pain or anything like that. So that means that they can be transmitting it to other people, and that's usually the danger behind these infections. If you can't tell, you're passing it on to others. But most of it includes things like your classic itching and irritation, kind of burning sensations during urination or ejaculation, that kind of thing, and that common issue behind discharge that is very common from a lot of the STIs that people are familiar with. So usually the other issue with trich is the fact that it becomes the precursor to having other sexually transmitted infections, and makes you far more propense to get infected with HIV. So this is one of the dangers associated with a lot of STIs in particular. Okay? So that's really one of the reasons why we look at this guy. Now we're going to move on to our second category. As you can see down the list of classification, we're moving into a new group called the Alveolets, or phylum Alveolata. And so phylum Alveolata, the term that probably most of you are familiar with already, is the term alveoli, which is the same term that you all learned, especially in AMP, when you look at the lungs. The alveoli are these little gas pockets, these little pockets of air. And so for humans, in terms of our lungs, that's what helps us kind of capture air and store it and exchange gases. But the same thing, it has the same meaning in terms of these protozoans, that it means a little gas pocket, a little bubble, if you will. And so these organisms, these alveolets, more than anything else, have these little pockets associated at the sides of their membrane or internally. And so that's where they get their name. Now, the other unique thing about them is that their mitochondria have this kind of rough, very tube-like shape, so hence the term tubular. And so within the pockets, the double membrane that everybody is familiar with about mitochondria, you have these cool little formations inside of them as well. Now, this is one of the reclassifications. This is where you kind of want to start paying attention versus what is in the lab. So we'll probably kind of compare them in a moment. Is that this has classes and subdivisions from phylum Alveolata into the three groups that have been originally described as phyla, which used to be Ciliata, Apicomplexa, and Dinoflagellata. They're no longer phyla. They're now their own subdivisions under the alveolets. And so this is Ciliata, Apicomplexa, and Dinoflagellata. So that's where we're going to go next, and we're going to use the lab manual to kind of go along and fix those to make sure we have the right information. Okay, so as we were mentioning with the alveolets, phylum Alveolata, they possess these pockets of air, very similar term that we use in the lungs. They have these modified mitochondria, and the current reclassification that occurs in the lab manual from being phyla that have become classes. These are ciliates, apicomplexans, and dinoflagellates, under classes Ciliata, Apicomplexa, and Dinoflagellata. So here, what we're really interested in doing is knowing some of the representatives under this particular phylum. So we go class by class. So looking at the very first class, which is class Ciliata, in this case, these are all chemoheterotrophs, but one of the unique things about them is that they happen to be binucleated, or they're binucleate. So what that means is that they have two nuclei in which one of them is slightly larger than the other. And these are usually referred to as a macro and a micro nucleus. So a big one and a small one. That's really what it's kind of telling us. So under there's three particular organisms that are important, and two of those pop up in our lab as well. So the very first one that we have in here is Watercella. Now, Watercella, one of the reasons why we see it, even though we don't usually play with it in the lab, is that this happens to be one of the few model organisms that we've studied so much when we discovered this field in microscopes. So this is probably one of the ones that is oldest in terms of being described and how much information we really know about it. Right? What's unique about them is that kind of bell-shaped structure that you see as part of its body, and that they have this cool little stalk that allows them to kind of attach themselves to things. This is that myoneme that you're seeing there. All right, so it's a really, really kind of unique structure, unique organism. I'm sorry, not unique structure. And so we kind of talk about it simply because it is one of the few model organisms that we have that kind of let us to understand this field altogether. And that's about it. Normally, when people are in general biology classes, they usually get a little bit of pond water and they kind of go find these guys, and they're relatively easy to find under a microscope under just scanning power. They're really quickly to kind of establish and figure out and see their bodies moving. And what's really neat about them is that little myoneme kind of squishes like a little worm, and it kind of almost crawls with that myoneme itself. So it's kind of an interesting way of motion. So remember, we classify these guys based on motion, right? Now, one of the ones we definitely want to know about, which is one of the organisms that you're going to be looking into in the lab as well, so that's why you saw the color change, is Balantidium Coli, which happens to be important for us. Notice that you can see the little hair on it, kind of the little bristles, if you can see close to the image. So that's the ciliate part that we're talking about, the ciliata portion, right? But we're interested in, because of it causing a specific disease called balantidiasis, or also known as balantidiosis. Either one of them really works, it doesn't matter. And this is a intestinal infection of the colon more than anything else. And as I'm pretty sure, probably the most common thing that most of you are familiar with is anything that infects your colon usually ends up having some sort of really large amounts of GI distress. What we do know about this bad boy is usually it's common in pigs. So this is probably our primary source of infection. But in terms of what it is, it's kind of persistent diarrhea, lots of kind of gut pain or abdominal pain, if you will. And every now and then it can get bad enough that it pokes holes inside your colon. So that's really where things can get really bad. This guy is really endemic in most tropical zones. So that means anytime you travel to the tropics, this guy is kind of a concern in terms of the water. So you may want to kind of resist the temptation of just drinking water from a pond if you need to, that kind of thing. So that's one of them, right? That's one of the other ones you're going to be looking for under a microscope. And then the last one that we have is probably the model of all time. This is the one that has been described the most. This is the one that has been studied the most. And this is the most well-known protist altogether, not even just under the ciliates, just all together, is Paramecium or Paramecium caudatum, which, as you can see the little hairiness behind it when you see it under the scanning electron microscope. And it's been studied enough to truly kind of observe its reproduction mechanism, and you can actually see the patterns that the macro and the micronucleus undergo, which is this very, very interesting kind of combination of sharing the micronuclei, and then ultimately those micronuclei will kind of fuse together and make several macronuclei. It's kind of a very interesting kind of pattern behind it. You don't need to know that, so I'm not going to ask you that. So relax on that. But this is one of the things that have been really, really well studied, and this is kind of our example or our model organism in terms of protozoans. So keep that guy in mind. All right. So that's the ciliates under Alveolata. So we're still under the phylum Alveolata itself. Now, when we enter the second class under Alveolata, so these are the apicomplexans, the name actually is kind of interesting to kind of derive in this case, simply because the term derives from the term complex and the fact they have this very unique structure called an apicoplast, which really what it is this kind of mini little drill that is used to penetrate the host. In other words, it gains access to the actual host by kind of drilling and poking a hole through it. So one of the things you can kind of deduce from that fairly quickly is that it's parasitic. So that means it can actually get into the organisms themselves. And so here, we're actually going to look at a couple of different organisms that are kind of medical importance more than anything else. And we will take out a couple of them in the lab as well. Okay. So let's go into these examples over here. So probably the first one that everybody should be familiar with is Plasmodium Falciprum, which for those of you who are not familiar with this one, probably the most infamous disease of all time, simply because of the numbers. There's really just no other disease that kind of overwhelms the planet is malaria. And so Plasmodium falciparum happens to be that organism that causes it. So these guys are all obligate parasites of virtually all animals and pretty much our insects as well, so which are included under that group, right? And so for us, they like to kind of hang out inside our blood cells and our bloodstream, and they'll introduce these cooler trophozoite stages, which is what you're seeing inside the our blood cells in there. And so they'll basically rupture our blood cells, which is what leads to the disease of malaria that you're familiar with. Now, for those of you who are not familiar with it, this particular parasite is transmitted through mosquitoes, specifically female mosquitoes, which are the ones that do the biting. And the actual classification of the mosquitoes is known as the Anopheles mosquitoes. And pretty much, like I said, there's nothing really I can exaggerate about this. This is the disease of the last few centuries for us. This is the #1 disease on the planet. So have fun with that guy. Now, the other one that is kind of interesting to kind of learn about is a disease called crypto, which occurs from an organism called Cryptosporidium. And in this case, Cryptosporidium, more than anything else, also another GI type disease. And this guy, what it likes to do is kind of hide inside the folds of the intestinal wall. So that those little folds that you have within your intestine, that's kind of where it kind of hides itself. And so that's the term crypto. So this idea of hiding more than anything else, that's where that guy goes. And the same thing, it's usually spread through drinking water, even in public places. So your classic, water fountain kind of thing, that's where you can kind of get it most often. And it's probably, if not probably the second leading cause of diarrhea in the planet. So most waterborne diseases, it's either this guy or cholera. So this probably is either #1 or #2, depending on the numbers each year. And then probably the last one and the third one that we'll be definitely looking into the lab as well is Toxoplasma, or specifically the organism more than anything else is Toxoplasma gondii. So this one kind of receives probably the infamous notoriety simply because of the fact that it's usually transmitted through cats. And so for those people that are kind of cat fanatics, this is where that disease kind of comes into play. Now, the disease is called toxoplasmosis, for those who are familiar with that guy, right? And even though it can be kind of transmitted through kind of undercooked meat from certain animals, typically it's because of having cats more than anything else, and usually from their fecal samples, either in the soil or changing out the litter box or something like that. That's usually where that guy kind of gets acquired. Now, some of the diseases, the issues that can actually cause, most of the people are kind of asymptomatic, but there are certain risks. So there can be certain issues in which it can get to your eyes, and it can lead from anything from eye pain to sensitivity to literally lots of watering or tearing of the eyes, and then you lose vision. So that's usually one of the more common ones, and until you kind of progressively lose your sight. So that's one version. And then anybody that usually has some sort of immunocompromised system, so weakness because of another disease, they can become very immunosuppressed and kind of can relapse into toxoplasmosis. And probably the one that most people are familiar with is certain neural kind of deficiencies occur because of a heavy load that can occur in the brain. Now we'll talk about specifically what's unique about toxoplasmosis once we deal with the lab portion behind that. So we'll leave the apicomplexans and enter the third of the groups of the alveolets, which are the Dinoflagellates or class Dinoflagellata. And here, this unique group happens to be all photosynthetic. So yes, that means that they're producing their energy from being under the sunlight. And they are biflagellated or biflagellate, meaning that they have two flagella. But what's unique about the flagella is that one of them is the one that kind of allows it to propel itself forward, and the other one allows it to kind of swim sideways and do some really cool turns. So the second flagellum kind of wraps itself around its body. Okay. Now, this guy composes pretty much the largest proportion of the plankton out there. So it is part of our ocean and part of our life cycle more than anything else for everything. Again, since it's photosynthetic, it contributes to our air cycle as well. And what's really neat about them is some of them kind of glow automatically simply because they bioluminesce, but a lot of them are also red colored, hence the term rotopigmented. And so when they grow in high amounts or in enough or sufficient amounts in certain bodies of water, they also have a tendency of also turning the water red. And so this is probably one of those other common occurrences that you'll hear in terms of newsworthy kind of information, in which you'll hear that water for some reason also turn into the color of blood. So that's where this guy comes into play. So let me kind of show you the structure behind this guy. Like I said, it's biflagellated, so that means it has one kind of flagellum that allows it to propel itself, the other one allows it to do some really weird kind of turns behind them. So that's the infamous site. As you can see, this is where that kind of blood color that occurs in the water as well. This is another one of those other organisms that cause that. Now, as opposed to the archaeans that we had talked about in the last topic, these ones actually happen to be toxic. So because one of the things that they create is something called a cottonotum toxin. The term means chain in case you're for the term. They also cause another toxin or create another toxin called the breve toxin. And both of these are neurotoxins. So that means that if you happen to be swimming in these oceans or bodies of water, whether fresh or salty, within just a few minutes of either swallowing a little bit of the water or it getting into your nose and hitting your brain, you most likely end up passing out. So this is why one of those concepts of seeing the water and people relating it to disease has this kind of historical or almost nearly biblical association with that particular organism too. All right, so now we'll leave the second phylum of the protozoans. So we left the alveolets, and now we're entering the third phylum of the protozoans, which are the cercozoans. Now, the cercozoan group here will enter behind their very high amount of similarity to the radiolarians as well as the amoebozoans. So we'll kind of piece these guys together and kind of show you the examples of how they move, what they do in terms of their overlaps more than anything else. So the next three groups, with one tiny little exception, which is really entertaining, all of them are going to have very, very similar properties and similar information that I want you to know.